Welcome to Nikolay's genetics lessons, and today's problem is a certain X-linked dominant disease in humans occurs when there are more than 40 repeats of the trinucleotide CGG in particular gene. Shown below are the outline of the pedigree of the disease described above and the representation of the gel showing PCR amplified fragments detecting the number of CGG repeats. The DNA corresponding to each individual is directly below his or her place in the pedigree. Based on the information you have been given, fill in the pedigree to show the sex of the individual as well as the phenotype affected versus unaffected. One more time, let me resume. So we know that this is X-linked dominant genetic disorder, and that means that genotype of the male would be, for example, X and Y, and of the female would be XX. So that means that males would have only one allele, so would have one band on the gel, and females may have two alleles with different size, so they may have two bands on the gel. So now let's analyze. In the first row we see only one band. That means that this is a male, or it can be a female with two alleles of the same size. But let's take a look here. So this is person 1, person 2. Person 2 has two alleles. So definitely in this couple, person number 2 is female. So let's put this sign here. And person number 1 is a male. So would be they affected or not? Take a look. This band is about 23 repeats. And we know that this genetic disorder manifests itself if greater than 40 repeats per allele. That means that this male is phenotypically normal. What about this female? One band corresponds to the 20 repeats, and another band corresponds to the about just a little bit smaller than 42 repeats, so we'll say 40 repeats. This is dominant genetic disorder, so because it's 40 or below, still phenotype of this female is going to be normal. Now let's analyze the children. Child number 1 have two bands. So this have to be a female. So this is have to be a female, and both bands are less than 40 repeats. That means that this female is phenotypically normal. Looking at the second lane, we can say that this is going to be female because two bands here and one band more than 42 repeats. That means that this is going to be female, but this female is going to be affected. In next lane we see only one band. That means that this is going to be a male because male has only one X chromosome. So this is going to be male. And what we can say about this band, it is bigger than 42 repeats. So that means that this male is going to be affected. Next, child number 4 is female. Here's a sign for female. And both bands less than 42 repeats, so phenotype is normal. Child number 5 is a male. So is a male and going to be phenotypically normal. Child number 6 also male, has one X chromosome, and unfortunately this chromosome has allele which is greater than 42 repeats. So this is going to be affected male. And next we have female, two bands and one band over 42 repeats. That means that this female is also affected just like her brother. And the last one is going to be male, and this male is going to be phenotypically and genotypically normal. So now we have a full pedigree of this family. We know phenotypes of all the individuals here and also the genotypes. And the last question which might bother you, why these children have the alleles of the different size if we compare with parents. Why not all of them have only this size, this and this, which we can find in parents. During meiosis, DNA polymerase, when come to such fragments where we have stretches of the same trinucleotide which repeats for multiple times, may slip and add extra trinucleotides. So this is what happens in this type of genetic disorders. And this explains why children may manifest this genetic disorder, those parents may not have it. And this is all for today. Subscribe and see you in the next video. Goodbye.